Hello, Biology 100, and welcome back to lecture for this week. This week we are covering Chapter 12 on the origin and diversification of life on Earth. Last week we talked about Chapter 10, all about evolution and where the theory of evolution came about and how all of those terms and terminology apply to evolution. Now we're going to take it one step further and talk about how Earth's diversification actually came to be based on all these evolutionary concepts. So, learning objectives for this week. We're going to talk about how life on Earth initially was formed. We're going to identify and name a species in terms of how we actually do that. We'll talk about the biological species concept and how that tells us what one species is and what a species is not, and also some pitfalls with the biological species concept and how the morphological species concept kind of helps us out with that. We'll talk about what is a phylogenetic tree and how we actually construct those. And then we'll talk about how your analogous traits and your homologous features factor into phylogenetic trees. We'll then compare and contrast microevolution and macroevolution and what those actually mean, as well as how adaptive radiation and extinction come into play with microevolution and macroevolution. Finally, we're going to understand how the three domains are current ways of classifying biodiversity in Earth. Okay, so the chapter starts out talking about how your organic molecules actually came from environments that didn't allow for life. So the Earth formed about 4.5 billion years ago, and the Earth itself was very volcanic and very turbulent, so not a lot of suitable conditions for life. The atmospheres were very gassy with things like ammonia, things like hydrogen sulfide, carbon dioxide, no real oxygen available for organisms to exist off of. Additionally, very stormy, very volcanic, so just very turbulent conditions all around. And in order for life to persist, there are two big categories that have to be obtained for something to be considered life. The first is that the organism, whatever it may be, has to be able to replicate, has to be able to make more of itself, whether that's through sexual reproduction or asexual reproduction, it doesn't matter, but it has to be able to make more. The second characteristic it has to have is it has to be able to utilize energy in some kind of fashion. It has to have a metabolism. So if it doesn't have these two characteristics, it is not considered life. So there are three phases in the sort of theory about how life came onto Earth. So as I mentioned, very volcanic, very turbulent conditions the Earth was undergoing. So no real oxygen. But it was believed, based on our evidence, that the organic molecules initially formed in these things called little warm ponds, basically these very heated pools of water on land. So phase one for the initial formation of life is forming small molecules that actually contain carbon and hydrogen. So how this was actually proven was through an experiment. So Stanley Miller and Harold Urey were a professor at a university and a graduate student, and they basically devised this four-step experiment to show how these little warm ponds actually created these organic molecules. So what they did is they took a flask with water and they put a bunch of gaseous compounds in it that were believed to be in the atmosphere on prehistoric Earth. So we had hydrogen, we have methane, and we have ammonia. They put those all in with a flask of water, and then their second step is they put a bunch of sparks into the flask itself to simulate lightning, so those turbulent, stormy atmospheric conditions. Afterwards, they then cooled the flask itself to form precipitation, sort of the condensation of the atmosphere, as it would have happened on prehistoric Earth. And then lastly, they examined those contents. And what they found was that not only did they have organic molecules, they also had found the presence of 5 different amino acids. And we know amino acids are the building blocks for our proteins, ultimately. So we have some proof of our first step. So that is phase one, producing these organic molecules like our amino acids. However, as I mentioned a minute ago, we have to be able to have these molecules or whatever it may be replicate in order to satisfy that first characteristic of life. So ultimately, the ability of self-replication and containing information came after these organic molecules came about. However, the problem is that for this to occur, for this replication to occur, we have to have energy, some kind of a reaction. So researchers thought that we need some kind of molecule that had catalytic properties. What that is an enzyme. And we didn't really talk about enzymes because we skipped over the biochem chapter. But an enzyme basically goes into a reaction and it lowers the amount of energy that you need to make the reaction happen. You have enzymes all throughout your body helping you pretty much perform every reaction possible. So breaking down food, respiration, you name it, there's an enzyme involved with it. So to replicate, we need some kind of enzyme-like molecule. And this came about through phase 2. So to be able to self-replicate, we need that catalytic molecule, that enzyme. And this is where it was believed RNA came into being. So ultimately, RNA is able to catalyze reactions necessary for replicating our nucleic acids. And with this RNA, we were able to replicate our organic molecules. So those amino acids, we're able to make more amino acids through RNA. And we know this is true because when we talked about transcription translation, we know that DNA codes for RNA, and then RNA is able to make proteins. So we have evidence of this from transcription translation. So ultimately, our RNA may or may not be considered the precursor to life because it's close to be considered living, and it's able to replicate the cell, or replicate the organic molecules. But ultimately, we don't yet have the ability to metabolize energy yet. So that is where phase 3 comes into being. So phase 3 is all about the development of a membrane, which allows the cells, quote unquote, to have metabolism. So as we talked about in the first unit, a membrane is a semi-permeable barrier that separates the inside of a cell from the outside of the cell. Semi-permeable being that it only allows certain things into the cell and certain things out of the cell. So it's sort of like a gatekeeper. And ultimately, by having this semi-permeable property, we're able to make metabolism happen because we can allow certain chemicals to come into the cell and certain chemicals to come out of the cell to ultimately catalyze reactions. So now we have our organism that has organic molecules, so in this case, amino acids. We have some way to replicate them with the RNA. And we have the ability to metabolize energy with the formation of a membrane. So ultimately, we have checked both boxes for life through these 3 phases. And the first life on Earth was considered to be your ancient prokaryotes. They satisfied both those major conditions for life, and we also have some supporting evidence that they were the first organs we have. The first one is that the age of the rock that they existed in was reliably determined through our fossil record. Additionally, the size of the actual impression they left in the fossil was found to check out with the records we have. And finally, with radiometric dating, which you learned about in the previous chapter, the ratio of isotopes is more like fossil organisms we have, or more like the fossil organisms of the prokaryotes and the actual rocks itself. If you remember with the radiometric dating, we are taking the age of a rock that has a fossil versus the age of the rock that doesn't have a fossil. And from that, we can parse out when an organism actually came into being. So ancient prokaryotes are the first believed organism on Earth. So just to recap all that, we have the 3 phases. So phase 1 for having life on Earth is the formation of our small molecules containing carbon and hydrogen. So our amino acids contain both of those. The second phase is the formation of our self-replicating information-containing molecules, which is RNA in this case. Then phase 3 is the development of an actual membrane to encompass the organic molecules and the RNA, making a sort of capsule, so creating an actual cell. So those are the three phases for having our first organisms on Earth. So now we're going to move on to the next section, which is all about what an actual species is. So what you'll notice, and can be tricky for species, is that in some cases, species can look completely different and actually be part of the same species. You would think all species are going to look necessarily the same. In this case, we have on the left, we have a human male and a human female being of different ethnicities and being of different genders and having different features, but ultimately they are the same species. However, on the right, we have organisms that look very similar, such as this Persian buttercup and the pink rose. They have the same petal structure and the same color. However, they are not the same species. Same idea with our second example. We have our Bornean orangutan, and we have our golden lion tamarin. Look very similar in terms of body size, hair, structure, all of that, but not the same species, happen to be different. So how do we actually define this? So we have what's called the biological species concept. And the biological species concept basically says species are natural populations of organisms that can interbreed with one another. Basically, they can mate and they can produce offspring. The offspring have to be viable in some case, or the offspring have to be viable in order for a organism to be considered a species in terms of interbreeding. So, for example, if you had a giraffe mate with the elephant, that's not going to produce viable offspring. Even if it could occur, it's not going to happen. So the organisms have to be able to interbreed with one another and actually produce viable offspring. So if you can't interbreed with organisms in your own group, this is what's referred to as reproductive isolation. Basically, you are separated in some capacity from reproducing with another organism. So with the biological species concept, we have reproductive isolation with two populations, and this can occur through either physical separation or it can occur through genetic separation. And we'll get to those steps in a little bit. But like I mentioned, species have to be able to produce viable offspring with one another in order to be considered species. So in this case, on the right, we have what's called a zorse, and that is the offspring of a horse and a zebra. So technically the horse and the zebra are able to mate and produce offspring, but however, this zorse is actually sterile. It can't actually produce offspring itself. So it's not really considered a species. It's sort of a weird hybrid. And with species, we have certain barriers to reproduction that prevent basically every possible combination of organisms existing with hybrids. We have what's called prezygotic barriers, and we have what's called postzygotic barriers. Your prezygotic barriers have the prefix pre, which means before, and then zygotic, we know, is zygote, which is the fertilized egg. So that's when the sperm actually goes into the egg and fertilizes it and then makes an actual embryo. So prezygotic before the zygote forms, and then postzygotic, post is afterwards the zygote. So postzygotic is after the zygote has been formed. So with your prezygotic barriers, these are factors that make mating impossible. So either the sperm can't meet the egg, or ultimately there's some kind of different anatomical structures that don't allow for mating. So there can be obvious physical differences between species that don't allow the act of mating to occur. There can also be differences in sort of social rules or courtship rituals. So certain species may have mating rituals that initiate mating between male and female. Some species may not. So if one species doesn't have a mating ritual, but the other one does, the species that doesn't have the mating ritual isn't going to understand these social cues to go on to reproduce. So prezygotic barriers, all about before the fertilized egg. With your postzygotic barriers, these are factors that occur after fertilization, but ultimately prevent the production of fertile offspring. So they may exist, but they may be infertile or they may just die. So another example we have with our post-zygotic barriers, and this is a mule. So the mule is a hybrid of the donkey and a horse. However, the mule itself is infertile. It can't produce offspring. There are other cases, additionally, with hybrid offspring where they just don't survive very long. Like they may not even make it to a year of life. So while technically it is feasible, quote unquote, it's not necessarily practical because the offspring just dies anyway. Okay, so those are our pre and post zygotic barriers. So now we're going to talk about how do we actually name a species. So most of you are probably familiar with species names just generally from your reading or from history class. You probably know things like Homo sapien or Canis familiaris, so that'd be human and dog respectively. But the actual breakdown of the name came from Carolus Linnaeus in the mid-1700s, and ultimately he came up with a classification system to name species in two parts. So the first part is called eugenus, that's the first word of the species, and then we have what's called the specific epithet, or the species of the species, and that's the second word. So taking this example with Homo sapiens, those are humans. Homo is our genus, and then sapiens is our species. So think of it like a first name, last name scenario. So Homo would be our first name, that'd be our genus, and then sapiens would be our last name, that'd be our species. So when you name a species, you always need both parts. Okay, so let's do an example, or another example. Here we have a zebra on the right, and using the Linnaean system, we can go from the highest order of classification all the way down to our species. So we have 3 domains, and we'll cover these more in depth at the back half of the lecture, called your bacteria, your archaea, and your eukarya. So up here at the top with our zebra, we have 3 domains. So bacteria, archaea, eukarya. Eukarya is the presence of a nucleus, so it's a eukaryotic organism, which zebras are. We then go one step more specific. These are kingdoms. This was the old-fashioned way of classifying organisms that actually was the main way we did it when I was a kid. So in this case, zebras are animals. And then we go to our next step, which is our phylum. It's phylum Chordata, which means it has the precursor to a backbone or a vertebrate. We then go to our class, which is the next level, which is Mammalia, so it's a mammal. We then go down to order, family, and then we get to where we are really concerned with, which is genus and species. So genus is Equus, and then the species is Quagga. So the full species name is Equus quagga. So you encompass the genus, which is the first name in this scenario, and the species, which is the last name in this scenario. So you have genus first name, species last name. But when we talk about a species, we have to use both the first name and last name scenario. So for our purposes for Biology 100, what I really need you to focus on is that domains are our broadest category that encompass all of our big groupings of organisms with bacteria, archaea, eukarya, and that genus and species are at the other end. I don't need you to know the phylum, class, order, family, etc. I need you to focus on the broadest category, the domain, and then the most specific with the genus and species. Okay, so with our biological species concept, it helps with a lot of our scenarios for naming species. However, there are a handful of shortcomings we have with naming species because there are some organisms that kind of break the rules a little bit in terms of the rules we set in place for naming things, but also just the way things work in the real world and ecology and the environment, things don't always play out as nicely as we'd like. So some things or some shortcomings of the biological species concept, there are 5. So we'll go through each individually, but to highlight them all, the first one is classifying asexual species. So that's species that just can reproduce by creating a copy of themselves. Issues we have with the fossil record, Ultimately, determining transition points from one species to another can actually be a little more tricky. Classifying what's called a ring species, and then classifying what's a hybrid species, so a mix of two organisms, like the zorse or the mule. So the first area of shortcoming we have is classifying an asexual species. So for a sexual species, we have some kind of fertilization. We have the egg meeting the sperm, either in utero or outside the utero. And that involves partners and courtships and breeding and everything. However, with asexual species, something like bacteria can just make a copy of itself and reproduce like that. So there's no real identity of reproductive isolation. So bacteria, it doesn't matter if it's separated from a bunch of other bacteria. It's asexual. It can just make a copy of itself whenever it feels like. So it kind of breaks that rule in terms of reproductive isolation. Next is ultimately difficulty is classifying a fossil species. So because the fossil record is not as robust as we would like, we don't necessarily know if organisms were reproductively isolated, if they had different structures or sizes or shapes in terms of reproductive morphology. We don't really know because, again, we're lacking a lot of evidence. Ultimately, because of the fossil record, we don't necessarily know when one species necessarily changed to another. We don't know that tipping point. Was it gradual? Was it very quick? We're not sure. For some examples, we have better evidence, but for many, we don't. So with the fossil record, we can't necessarily always ID that transition point. When did it exactly occur? When did species A become species B? Next, we have difficulties in classifying what's called a ring species. So this is going to require a little bit of an explanation to get behind. But the idea behind a ring species is that you have a population of organisms that could, of course, breed with one another because they're all the same species. But ultimately, they get separated by some large obstacle, and they go. Part of the species go around one way, and part of the species go around the other way. And when they meet on the other side, so much evolutionary time has passed, they actually are separate species. So the example we have is your greenish warblers. And over here on the right, I have a picture of the initial population of greenish warblers that I'm circling. And this population exists at the base of the Himalayas, the Himalayan mountains. So a very big obstacle that the birds can't go over. They can't exist they can only exist at a certain altitude. So once they get too high, they won't be able to survive. So basically, they're stuck at a certain altitude around the mountain. So they can't go over the mountain, they can't go under the mountain. So they're going to, of course, go around the mountain as their population spreads out. So part of the population of greenish warblers go to the right, and then part of the greenish warbler population goes to the left. So they start to spread out around the mountain as they expand. Ultimately, we have potentially hundreds of thousands of years pass, and that's a lot of time evolutionarily for allele frequencies to change based on different environmental conditions. So the right population may start becoming genetically different from the left population, and they're, of course, only reproducing with one another. So individuals from the left population are only reproducing with their left side of the mountain populations. Same with the right side. Ultimately, they'll keep expanding until they get back around the mountain and they'll meet up. However, because so much evolutionary time has passed and so many genetic differences have occurred, these species actually cannot reproduce with one another. So the left population is now a completely different species, and the right population is now a completely different species. So these are what's called ring species because they basically go in a ring around some sort of obstacle and they'll meet up. So on one side of the ring, they are the same, but on the other side of the ring, they are completely different when they meet back up. So because we have this big transition, it's kind of difficult to know exactly if this left population can still produce with our initial population, if this right population can produce with our initial population, if the right population can produce with our final population of right side of the mountain birds, or if our left population can produce with the left side of the mountain birds. So it gets very tricky about where we draw the line with where does the reproductive isolation start and end. Okay, so the last big shortcoming we have for the biological species concept is hybrids. So hybridization is when closely related species, but not the same species, produce an offspring. So your mule and your Zorus are the examples again. And while the hybrids may be able to exist, they can be either infertile or they can potentially breed with either parent to produce offspring. So if species A and species B are really close related and they produce an offspring, let's say that offspring is species C, or sorry, let's say it's species AB. So parent A, parent B of different species produce offspring AB. AB can now reproduce potentially with A or B or others of itself. So we have 3 possible combinations of different breeding choices they have. So does that mean that they're all now the same species? Does that mean they're subspecies? We don't really know. So it's kind of hard to draw lines again with this. So one way we try to combat this is with what's called the morphological species concept. So with the morphological species concept, we are basically now basing species based on physical features, things like body shape, body structure, bone structure, things of that nature to sort of classify things instead of just who can breed with who. Okay, so the actual process by which species arise is called speciation. Pretty straightforward definition. It's just when one species splits into 2. So, for example, with those greenish warblers, one side of the mountain, they were one species. The back side of the mountain, they were 2 different species. And there are 2 phases for speciation. The first one I kind of touched on a little bit. It's called reproductive isolation. That basically means that individuals of a species aren't able to reproduce with another individual of the species for either geographic or non-geographic reasons. The second reason is called genetic divergence. And this is basically when 2 populations evolve enough and they have enough physical or behavioral differences that they can't produce. They can't reproduce. They've had a bunch of genetic changes, whether that's through natural selection or mutation or some combination of both. So 2 types of speciation we have. The first one is called allopatric speciation. And the second one is called sympatric speciation. For allopatric speciation, that is when we have some sort of geographic isolation that results in the genetic divergence. So at the bottom here, we have 2 different types of ground squirrels, 2 different species, but ultimately they were once one population of the same species. So for here on the right, we have our initial population of ground squirrels right here, but we have reproductive isolation through a physical barrier. That physical barrier is a river forming. So over hundreds or thousands of years, that river forms through the habitat that the squirrels live in, and ultimately the squirrels can't cross over the river. So one part of the population is stuck on one side, another part of the population is stuck on the other side. Ultimately, left side can't reproduce with the right side because they can't reach each other. They are stuck on their respective sides. That is an actual physical barrier creating reproductive isolation. So over time, the left side just reproduces with the left side, and the right side just reproduces with the right side. Natural selection, mutations occur, and ultimately they have genetic divergence. They are now no longer able to breed with one another, even if they were able to cross the river. So allopatric speciation is all about geographic isolation when we're talking about reproductive isolation. Another good example we have is with our Galapagos finches. So on the mainland off the coast of Ecuador, we have a mainland finch. And ultimately, some of those finches migrate out of the population onto certain islands, and then enough time passes, and then through natural selection or through mutation, we have a bunch of different type of finches, such as the woodpecker finch, the large ground finch, small tree finch, vampire finch, and large cactus finch that all have their special traits that allow them to live on their island successfully. But since enough time has passed and we have a geographic barrier being the ocean in this case, they aren't necessarily going to want to cross it, or if they even could, they are now too different to reproduce. So that was allopatric speciation. Now we have what's called sympatric speciation. So sympatric speciation is when we have speciation without geographic isolation. So this is kind of a weird scenario because usually we would think that organs would need to be physically separated in order to not be able to reproduce. So this doesn't really happen a lot with our vertebrates. So like our animals, for example, like our ground squirrels. Usually if the same organisms are in the same space, they're gonna reproduce, assuming they're the same species. However, this is very common among the plants in the plant kingdom. And this occurs on a genetic level. So with our sympatric speciation, we can have two different scenarios that ultimately lead to individual species occurring this way. So again, remember, sympatric speciation is that they occupy the same geographic space, but they're still not able to reproduce. The reproduction barriers are a genetic level. So two ways this can happen. The first way is called polyploidy, which we touched on really briefly when we looked at genetics, mutation, and chromosomes. Polyploidy is basically having more chromosomes than are necessary. So we know that organisms are normally diploid. They have 2N or 2 sets of chromosomes. But basically during an error in cell division, organisms can actually not have their chromosomes separate during meiosis. So ultimately when they get fertilized, they have an extra pair of chromosomes. So polyploidy in this case for plants, one of our organisms had its meiosis occur properly. The other one did not have it occur properly. And ultimately, when it was fertilized, the organism had way more chromosomes than it needed to. Because of this, the polyploids are now not able to reproduce with non-polyploid plants, even if they're the same species. Polyploid plants of one species can only reproduce with polyploid plants of another species. So in that way, they're now reproductively isolated despite occupying the same space. The second way this can happen is ultimately when you have a hybrid form from two closely related species. So in this case with our plants, we have a species 1 gamete and a species 2 gamete. Those come together and get fertilized and produce some kind of hybrid. However, the hybrid in this case is not able to reproduce with any of its parent species. It can only reproduce with hybrids, with other hybrids, I should say. So now ultimately, the species hybrid is now kind of stuck in its own box. It can't reproduce with either parent that made it, either parent species that made it, only other hybrids. So in that way, it's now closed itself off reproductively in that case. That's the reproductive isolation. So recap, two ways that sympatric speciation occurs in plants. The first one is your polyploidy, and the second one is the formation of your hybrids. Okay, so I'm going to stop it there and pick up with part 2 in a minute.